My name's Professor Diane Fatkin and this is my colleague Dr. Inken Hutner, and we're from the Victor Chang Cardiac Research Institute in Sydney, Australia. And our research is focused on looking at genetic causes of dilated cardiomyopathy. So let's look at this echo. So here you can see a normal sized left ventricle, and in comparison we can see that a ventricle in a patient with dilated cardiomyopathy is very enlarged and the pumping action is substantially reduced. There are many causes of dilated cardiomyopathy, but genetic changes are known to be one of the major causes. And the big news story in the genetics of dilated cardiomyopathy is the role of titin truncations, and that's what our paper's all about. So Inken, what can you tell us about titin?
Yes, titin has been the focus of my research over the last couple of years, and here on the screen you can see a diagram of the titin molecule. It is a gigantic protein. It's the largest protein known in humans, and it spans the entire width of a half sarcomere all the way from the Z line to the M line. The two most important regions of titin are the I-band region of titin, which is important for the elastic properties of the heart, and the A-band portion of titin, which is important for the contractile properties of the heart.
So let's think about what this big news story was. In 2012, a next generation sequencing study showed that truncating mutations in the titin gene were present in up to 1 in 4 patients with dilated cardiomyopathy. Now this was phenomenal because it was equal to the prevalence of mutations in pretty much all the other disease genes all added together. And in a recent study of over 5,000 individuals, this very strong association between titin truncations and dilated cardiomyopathy was confirmed.
That's true, but the other interesting finding from this study and subsequent studies since was that not only patients with dilated cardiomyopathy carry those titin truncating variants, but also control individuals in the healthy population. In fact, up to 1 in 200 control individuals carry those titin truncating variants.
Well, this raises the real controversy as to how important titin truncations are as a cause of disease and whether they're sufficient alone to cause disease. So there's some progress has been made, and we now know that not all titin truncations are equal. And let's look at this titin molecule diagram here. You can see that many of the exons in this I-band region are spliced out and not used in all the titin isoforms. And it's thought that titin truncations in this area may not be all that important. Whereas titin truncations that occur in the A-band tend to be in regions where all the exons are expressed. It's thought that these may be more functionally important. But the titin location debate has recently been further extended. Inken, you were telling me about some work with zebrafish on this.
Yes, an interesting proposal has been published in a group that studies zebrafish with titin truncating mutations, and the authors of that study found that there is a novel transcript of titin termed the Kronos transcript, which is transcribed from an internal promoter of titin, and that transcript is a C-terminal transcript only. Hence, mutations that would be localized in the more C-terminal portion of the protein would affect both full-length titin as well as the novel Kronos transcript, and hence be more severe than N-terminal titin truncations, which would only affect the full-length version of the protein.
So that suggests that not all titin truncations are equally pathogenic.
Right.
But coming back to this question as to whether titin truncations are sufficient to cause disease, there has been some progress made with animal models. And Inken, you were telling me the other day about some work in zebrafish.
That's right. There's a couple of zebrafish models of titin truncation which show that in the homozygous setting, titin truncations are sufficient to cause disease, but that is very different to the heterozygous setting of a titin truncation in a human patient.
Well, that's where the animal models are particularly interesting, and there are heterozygous mouse models. And very interestingly, the mouse models don't show any baseline evidence of dilated cardiomyopathy, and they only develop contractile dysfunction when there's a second stress applied.
That's correct, and I think the real animal model that truly recapitulates the human phenotype, that is still in need to be discovered.
So the real question that cardiologists want to know is, what do they tell their patients with titin truncations? So what we can say is that if your patient already has dilated cardiomyopathy and a titin truncation is found, there's a good chance that that titin truncation is contributing to disease and that that person has what it takes with respect to background genetic factors or environmental or lifestyle factors for this titin truncation to be manifest.
Well, the other scenario is that titin truncating variants are found in the context of an incidental finding in a genetic testing scenario for a very different purpose. And it's unclear at this point what we would tell a patient with an incidental finding of a titin truncation, because we don't know whether that that patient would develop DCM in the future or not.
So in our paper we've highlighted some of these critical issues with respect to titin truncations. Really there's a lot more work needs to be done to sort this out and to give us real guidelines as to how to manage our patients with these variants.
That's right.